All right, so welcome again to our Excel educational webinar. It's 3 minutes past 2 and it's time to start. I can see that people are slowly joining, and I would like to introduce and thank Dr. Steffen Cosson. You need to unmute yourself, and let me just give you a little introduction to Steffen. So today we are going to talk about portfolio of SCIENION, and Steffen Cosson is the senior scientist at cellenONE. He joined in 2019 and is taking care of the spheroONE project. So he's actually the leading person for the spheroONE project. And this project, for what I understood, aims to develop and automate spheroids and organoids isolation. So I would like to thank you once again, Steffen, for your participation today, and we look forward to hear more from you.
Yeah, thank you for the invitation and the kind words. It's a great pleasure to be here and present our work to the audience. So today I'll show you like a brief overview of our portfolio at cellenONE, and mostly like highlighting some of the instruments that we have for single cell isolation, as well as some of the application our customers and we have developed with such systems. Then I transition a bit with our newest instrument that really was developed to really process spheroids, organoids, and so forth. So first, I'll just give you like a brief overview of our company. So we are cellenONE. We are a spin-off company from SCIENION, which is a global leader in diagnostic manufacturing based in Berlin. And what we do at cellenONE is really to transition the technology that was first developed for diagnostic manufacturing, for instance, precision low volume non-contact dispensing, for application in the biological world. Our mission is really to provide disruptive technologies, products, and services to better understand and master cell biology. Another point which is of interest is in 2020 we got acquired by PICO, formerly CellLink, which is now one of the world leader in bioconversion. So like using technology to really address biological questions and so forth. And we are part of this bigger group of companies with a lot of sister companies that is just listed here. So cellenONE is really developing instruments that allows our customers to really address some of the specific needs, in particular isolation of single cells with the cellenONE, and now also with larger cellular aggregates. And with the two instruments, you can really process, isolate, dispense for various applications, a large range of biological samples. So first, I will go through our flagship instrument, which is the cellenONE, which is an instrument that is really dedicated to isolation and dispense of single cells. So first, I'll just tell you briefly how it does work. So the principle behind it are threefold. So first, we have precision low volume dispensing, which is piezoacoustic based, and this allows us to very reproducibly, rapidly, and with high accuracy generate teeny tiny drops in the picoliter range. This dispensing unit is actually mounted onto a XYZ robotic axis that allows us to really dispense these teeny tiny drops literally on any substrates, be it like glass slides or various tissue cutter well plates and so forth. And we are very open towards the targets that we can use with the system. As you can see, we have a glass capillary that is transparent, and for isolation, we actually place that capillary in front of the camera that allows us to take an image before every single drop is generated. That allows us to track and identify objects within the capillary as they are ejected. To further explain how the system works, here is a small animation that really go through the whole entire process. So this is the cellenONE. It's our instrument, flagship instrument, and what you have in here is different parts. So you have first, you can use and mount it with different types of labwares. be it multi-well plates or custom-made glass slides ranging from 96 to 1536 well plates. The target holder for the well plates can be temperature controlled, which is often used. Now, what we're going to do is go into a single cell suspension, aspirate it into our capillary, and then we place it in front of the camera. That allows us really to look at the very tip and be able to take an image before we generate any single drops. What we have also is a mechanism, I just pause it right now, where we actually determine an ejection zone which corresponds to the volume of the next drop that is to be ejected. On top of this, we had a buffer zone, which is called the sedimentation zone, that we use to take into account the possible sedimentation of object into the capillary. So what the system does is it takes an image, analyzes it on the fly, and decide automatically whether there is a single cell in that ejection zone or no cells. In the case there's no cells, the drop is discarded and collected in the collection tube. And when we have a single object, the robot axis will move the capillary on top of the next target well and shoot the drop with the single cell inside, ensuring that we have in every single well a single cell and nothing else. As I said before, when you have no particles or multiple particles, the drops are discarded and actually collected into a collection tubes, which can be addressed back. So you can go back to your collection tube and actually recover your suspension and reprocess it. Because we have an image-based system, one important aspect is that we can do measurements onto the object that are inside. So you can measure the size of your cells, you can measure their elongation, you can measure their fluorescent intensity and so forth. And we use that to really characterize the cells and be able, for example, to not isolate doublets of cells as seen in the next pictures. Every picture is saved, meaning that you can always go back to the image to track and have some kind of QC that allows you to really know exactly what was dispensed in each of the well of your target well plate. And so forth. So this is just a live video that shows you the process as it actually occurs. So it's fairly rapid. Typically, it takes you 3 to 5 minutes to fill an entire 96 well plate with single cells, and you can really use that to ensure that you have up to 100% of single cells dispensed in each of the well of your target well plate. Just as a summary and a recap, so we have these two distinct regions in the capillary. You have in green the ejection zone and in red the sedimentation zone. So only when you have a single object in this green zone, the ejection zone, would the robot actually move on top of the next target well plate and generate the drop, ensuring that you have a single cell dispensing that in that well. Whereas automatically the system decides to just not move and discard drop into the collection tube, would there be no particles, no cells, or multiple cells within the ejection zone or the ejection zone and the sedimentation zone. So as an example, here is just an array of single drops containing a single cells that were arrayed onto a glass slide. The cells were recovered from a mammosphere, so an aggregate of maybe breast cells, if you want. And here you see really like the accuracy that can be achieved with the system, where every single drop really contains these single cells. And because we have an image-based system, the system is not biased by the size of the cell. So you can really set the machines to isolate every single cells, wherever it is a small cells or larger cells, which allows really like to address the heterogeneity of your original sample. As I said, the system can be used to isolate and dispense a wide variety of biological samples, ranging from tiny bacteria, where we need to use fluorescence because they're too small to be seen in transmission imaging. You can isolate some nuclei, you can isolate some spores from plant cells, any type of mammalian cells, and you can even go with like fairly large cells such as cardiomyocytes. So with regards to application of the system, our main application from our customer perspective is cell line development, so cloning for example, for antibody productions or for selection of cells that were genetically modified using precision gene editing. And here is just how we do it. So on the left, what you have like is calculations where we display the efficacy with which spheroONE can be used to isolate single cells and generate single colonies in each of the well of your target well plates. So that is like ranging from 96 well plates, 384, and 1536. So the system is really compatible with any type of library. And you see that we can achieve very high efficacy of isolation of cells, up to 95%, all the way to 100%. And this is really striking difference when compared to standard liquid handling processes, such as serial dilution, where you are poised with Poisson distribution, where typically the yield you get of clonal outgrowth is only 25%. And because we are so efficient in the isolation, this allows our customer to use our system and really accelerate the whole process of cell line development. So typically, because you don't have to do serial dilution, all your wells are going to have a single colonies, and that allows you to really, within half of the time, to generate the colony and identify the best clones. Where typically using the standard serial dilution protocols, you would need up to 8 weeks to do it. Another comparison study that we did is to compare the cellenONE with atypical FACS. So in this particular assay, each system, so the cellenONE and the FACS, was given 1,000 abridoma cells. And out of that, what you can see in the graph is that using the FACS, out of these 1,000 cells, 533 were actually isolated into the, were just isolated. With the cellenONE, we achieved a slightly higher number with 664 ones. What is most striking is out of these isolation, only 75 wells gave rise to a single clone using the FACS, whereas 480 wells gave rise to a single colony. And this really highlights the fact that the dispensing used in the cellenONE, so these piezoacoustic precision dispensing, is very gentle with fragile cells, such as abridomas, whereas FACS is often poised with some low viability due to shear stress. Oh. Yes. Sorry. Recently, so earlier this year, we also launched what we call the cellenONE HT, which is a cellenONE that is being optimized for full automation. So what it means is that the cellenONE is equipped with a teeny tiny plate shuttle that can be addressed using a robotic arm that allows us to automate the entire bioproduction workflows. So for example, what happens in such a typical workflow is you would start with like a sample plate where you have your single cell suspension. That sample plate is fed to the cellenONE. The robot goes into, with the capillary into the first well, aspirate the single cell suspension, and then distributes and isolates a single cell in each of the wells of the N or the 1, 2, 3, 4, 5 target well plate that you want. Once you run out of sample, the system can get the sample plate back, you aspirate the cell suspension from the second well, and you repeat that process over and over and over. That allows you really like to have very high throughput cloning into your labs. Yes, that's all right. Another set of application that our customer use the cellenONE quite a lot is for rare cell isolations. So in the first example, what we have is liquid biopsies taken from patient possibly have cancer, and looking at isolation of CTCs, so circulating tumor cells from the liquid biopsy, so typically like a blood sample. The amount of CTCs you have in there is very low. So typically you would have 1 in 10 to the power 10 cells of interest. We collaborated with Sequence to get these liquid biopsies, and with Biolytic, that has an apparatus that allows you to enrich numbers of CTCs. With that Biolytic instrument, you can actually enrich your samples to have, like, let's say, 1 CTCs out of 1,000 or 10,000 cells. And this is sufficient enrichment for processing with the cellenONE. So from that enriched liquid biopsy, the cellenONE is actually capable of isolating single CTCs for your downstream application analysis. Is it just going them to test them, or single cell RNA-seq or genomics, and so forth. Another example is quite the opposite. So the UCSF, with whom we collaborated, are interested in recovering cells from cerebrospinal fluids. There, the issue is that it's a fluid that is poorly, have a very low content of cells. So typically, you have 1 to 25 cells in every mil. So it's not a lot. So what we do is we take that liquid biopsies from the cerebrospinal fluid, we concentrate the cell concentration using a simple centrifugation steps, and then we use that with the cellenONE. That allows us really to isolate these very rare cells very efficiently, and allow our customer or collaborators here to run downstream cells on these very rare cells, which is difficult to get a hold on using other techniques and things like that. Another application our customer have been very successful with over the past couple of years is single cell analysis, such as omics. Just to stress the usefulness of single cell analysis, just think that in your body, your tissues, your organs are made of a multitude of cells that are different from one another. So typically when you do a bulk sample, so when you do analysis of hundreds of cells that are taken from your tissue, the signal you get out of that analysis is typically the average of all these different cells. And this is really where it is important for us to really go for single cell analysis, because when you analyze single cells, you can actually define within your original tissues subpopulations. And this is very interesting because you can really address your biological question on the cells of interest and not just an average of various different cells and so forth. Just a heads up. As I said, so the cellenONE is a single cell isolation system that allows you to isolate and dispense single cells, but it is also a precision dispenser. On top of that, we have environmental and temperature control of the targets, and this is really at the combinations of the two things really allows our customers and us to develop entire dedicated workflows for single cell analysis and applications. And now what I will do is go through a couple of highlighted customer applications that was done using cellenONE. In the first case, here is a single cell whole genome sequencing, which is called the GLP+, which was developed initially in Canada, which allows them to really process lots of cancer cells coming from very different sources, typically from patients, and allows them to really sequence large numbers of cells. So right now they have sequenced above a million different cells from different patients, from different types of cancer and so forth. This particular process has then been implemented in multiple cancer research institute across the globe. Interestingly, if you look at the caption here for figure 4, if you recall, every single isolated cells, you have an image that is taken and saved, so you can track it back. So what these researchers here in this paper have been able to do is to correlate the sequencing. So typically, wherever the cells is tetraploid or diploid, and they could correlate that with a very specific morphogenic trait, where diploid cells are really round and nice looking, whereas the tetraploid cells, so the aberrant cells, would have a very distinct shape and contrast. What we also have done is developed a custom-made labware, which is called the cellenONE 3D4. So it's micro-well arrays in the format of a glass slides that allows us then to run a single-cell 3' RNA-seq in a nano-volume range. The fact that we do this in nano-volume allows us to have a better signal-to-noise ratio, so therefore having a better readout of your single-cell transcriptome. To illustrate this application, so what we have done is we did the comparative studies of the transcriptome of hepAIG cells, so cells from wheat product that are hepatocyte and typically used for ADME-tox assays for drug screenings. And we looked at the expression, the gene expression of cells grown in 2D compared to cells grown into single aggregates. And you see that there are a lot of differences between the two different gene expression in there. So we could look at a large variety of genes that are expressed in these different cells, and we could easily cluster them. So you would have really a cluster of cells that were grown in 2D and another cluster for cells in 2D, which really highlights the fact that cells grown in 3D would have a different gene expression pattern when compared to cells grown on a monomer, which is really interesting. Recently, so like last year, our collaborator at EMP Vienna, really with collaboration with my colleagues in here, were able to develop a workflow for single cell proteomics using a multiplex approach. So here what you have is, again, multi-well, micro-well arrays that are set into a glass slide format. In each of these teeny tiny micro-well, you can isolate a single cells, digest it. Then what you would do is add in each of these wells an isobaric tag that allows you to track the protein fragments from a single cells. You would then pull them 16 by 16 and run that through your HPLC and then for your mass spectrometry. And this is the first application for such single cell proteomics. Just yesterday, we launched a label-free process for single cell proteomics as well, in collaboration still with IMP Vienna and Broker. And here is just a different way that people can do single cell proteomics, where you don't pull necessarily the cells, but you do it cell by cells. And that's just based on different mass spectrometry devices from different distributors. Even more strikingly, so the Pacific Northwest University in Seattle, US, showed for the first time that you could do single cell multiomics analysis. So in this particular process, what they do is from a single cells, they were able to look at the gene expression as well as the proteomics, also the protein present in a single cells. And this is really, for me, very, yeah, it's hard to believe that they're able to do this. So it's a very nice applications, and it opens up for a lot of future applications and looking at multi-omics analysis of single cells, which is really eye-blowing. So just as a sum up for the cellenONE and just looking at the unique properties that it offers our customers. So you have accuracy, you are fairly confident that you will have a single cells dispensing each of your target well plate. And that is due to the image-based sorting that we provide with the system. Because the dispensing is so gentle, you ensure the high viability of your fragile cells or your cells of interest, which may not necessarily survive isolation for FACS. Because we have an image, you can always trace back your analysis to an image of a cells and really correlate morphological parameters with regards to the analysis you did downstream. It is compatible with any type of hardware, and also because we have the possibility to do precision dispensing, it really enables us customers to develop and to implement entire workflow into a single instrument, which is very nice because you don't need a liquid handler, a fax, and all of that. You have everything in a single instrument that does everything for you and can be programmed. Now we'll switch up a bit of the gear, and I'll show you some applications we have with the spheroONE, which is our instrument that processes cellular aggregates such as tissue biopsy fragments, spheroids, organoids, tumoroids, and so forth. The basics for the isolation of these aggregates is the same. So we have a precision dispensing technology, which is slightly different because we generate larger droplets. So for the spheroONE, we have a pressure-based system with a solenoid valve to just opens and closes. That allows us to really generate drops in the nanoliter volume, so 1,000 to 10,000 times bigger when compared to the cellenONE, with high efficiencies. Because the samples we process now are significantly larger than single cells, because they compose of thousands and thousands of cells, we had to enlarge the aperture of the capillary that we use. So typically, the current offering that we have is to have capillary ranging from 100 nanometer in aperture all the way to 600. And then we have a pressurized reservoir where you will need to store your samples, allowing you to process your spheroid suspension quite easily and maintain it sterile, because it can be prepared under a biosafety cabinet, put into the system, and then isolate. And we also have, as per the cellenONE, different optics and imaging capabilities with up to 4 different fluorescence channels. So looking at the workflow for the spheroONE, this is how it works. So typically, you can use any type of culture methods to form and culture your cellular aggregates. For example, overlay liquid, just agitating the cells, using these microarrays where you pellet the cells in the bottom of these microwells to generate your spheroids. What we need is for you to be able to take a suspension from that culture and then feed it into the spheroONE. And then what it will do is exactly the same. So we have the imaging that comes. You will look at your extraction zone, check that you have a single particles. When you have a single particles, it will move on top of your target well, dispense the drop, ensuring that you dispense a single aggregate in that well. Because it's image-based, as per the cellenONE, you can do analysis on the actual object. So check the size of the aggregates, whether it has a given fluorescent marker, and so forth. Once you have filled every single well of your target well plate with an aggregate, we believe that you have an assay ready plate. So you could be able to drug it, directly incubate it, and then run your readouts right away. So in practice, what can you expect from the system? So here's just an illustrative example. So you start with your spheroid suspension, which can be fairly heterogeneous in terms of size and shape, as seen from the picture in here and from the scatter plots with the rings in blue. You process that sample using the spheroONE to isolate a single aggregate in each of the wells of your target well plate. The dispensing is very gentle. We use very low pressure, which is significantly lower than what you would have into a fax. That ensures that the aggregates maintain their integrity and their viability. The entire process is highly accurate. And looking at the pictures you have here and from the scatter plots, where each of the pink dots that you see here is an actual isolated aggregate, you see that you can really select a subpopulation of your original heterogeneous populations. To further illustrate this, we took that same sample and we set the machine to isolate the aggregate based on a size gating. So in the top example, we set the gate to be fairly large. So we isolated every single spheroids that would have a diameter ranging from 100 to 500 microns. From the image and the box plot, you see that you end up with an isolated population of spheroids that is fairly heterogeneous, representing a bit the heterogeneity of the original sample. In the second example, we set the size gate for isolation to isolate everything that was having a diameter between 350 and 500 micrometers. From the images, you see already that we only isolated aggregates that are fairly large, and this is confirmed with the box plot. In the last example, what we wanted to show is really how stringent the system can be. And here in that example, we only isolated spheroids that would have a diameter ranging from 200 and 250 micrometers. You see from the image and from the scatter plot that you can really achieve a high homogeneity with regards to size of the sample that you isolated, which is really something you are looking for when you want to use these models for drugs testing or for any downstream application you may have with those. With regard to applications, so these 3D models are often used for drug screening more and more. Now you have roughly spheroids used in every kind of drug test, as they are more predictive and more representative of the physiology of the healthy tissues and diseased tissues. Now you also have the avenue of organoids, which are typically tissues and mini organs that you can grow on a dish, which are really very promising for modeling healthy tissues and diseased tissues. And you want to be able to manipulate them quite easily. So organoids are typically cultured in matrigel. So I must say that currently the spheroONE is not able to dispense matrigel. However, because our target plate order are temperature control, what we can do is pre-fill every single well with some matrigel, keep it at 4 degrees such that the matrigel remains liquid throughout the isolation process. And that allows us to really encapsulate, embed the aggregate inside of this matrix, as we can take it from the instrument, put it into the incubator. And that is really useful for passaging or growth of these very peculiar models in complex that are organoids tumoroids. Another application that we have looked at is advanced image processing. So in collaboration with some bioscience in Switzerland, we have shown that using a specific illumination setting that is dark field, where instead of shining directly the capillary with the light, we shine it with an angle such that we can actually collect the light that is diffracted by the cellular aggregate. Typically, what you obtain as a diffraction pattern for a full aggregate is an image of the aggregate that has an homogeneous diffraction pattern. Whereas when you shine a cystic aggregate, so an aggregate that only has a layer of cells and is empty inside, you can clearly see that you have a different diffraction pattern. That is because it's empty inside. So we believe that with time, we should be able to use these differences to be able to tease out and sort out, specifically sort cystic aggregates from four plain ones, which is very interesting for certain applications where you really want to be able to select these cystic organoids, especially in the organoid culture and selection of the best model that you can have. So as a short sum up with regards to the spheroONE, so as per the cellenONE, we are confident in our accuracy in single aggregate isolation in each of the well of your target well plate. Because the user can really select the parameters for which aggregate is or is not to be isolated, we can really achieve high homogeneity of the sample that is dispensing your target well plate, which ensure that you have homogeneous populations and the best sample possible to use for your downstream applications, such as drug testing or analysis and so forth. Because we start always with a suspension of aggregates, you can always pre-process it. For example, we do recommend to strain it to remove any cellular debris or single cells or dead cells that might be in there, and that just to ensure that within the drop you dispense in the well, you won't have any of these debris around it, and have a really clean plate to run your downstream applications. The system is also capable to dispense multiple aggregates per well. This is really defined by the user, so you can dispense a single aggregates, 3, 4, 5. It's really up to you. Just need to know that the limitation is still done one by one. So if you want to put 100 in a well, it will take some time. And next, we are compatible with the most prominent means of culturing organoids, so we can embed aggregates inside image gel. We have the possibility to look at morphologies and process the sample with high viability and high efficiency. With this, I will thank our customers. I think it's really very nice, and we are very proud that with our instrument, our customer were able to develop some of the applications that were highlighted in this presentation, and we would not be able to show you this without them. This is the team here in Lyon, and with that, I'll thank you for your attention. I hope you enjoyed the talk, and I'll take your questions.
Yes, thank you, Steffen, for the nice presentation. So, and I just want to add that we have a new installation here in Prague at the BioSev, so we are really happy to see the data generated by this young team. And yes, of course, so now it's time for question, and I can see a quite nice Polish participation. So don't be shy and ask Steffen any questions. So we are happy to answer.
Look into the chat. Yes. So with regards to, so one question is a few words about CRISPR application on cellenONE, please. To my knowledge, we have only a few customers that are interested into cloning from CRISPR modified cells. I don't have any data that I can share right now, but I'm sure if you give I'll look into our application, I'm sure I'll find one and I'll be able to do that. But typically what happens is people use that to isolate single cells and look at the best expressing clones. That's all I can say.
Okay, thank you.
No,
just one of my customers is dealing with yeast.
Yes. So the cellenONE can process bacteria such as yeast. The difference is because they are so small, the isolation is typically done using the fluorescence channel. So you need to be able to label your yeast with a fluorescent marker to be able to isolate. We are also working currently on dark field illumination, and this is work in progress right now with my colleagues in the microbial team.
Okay, thank you.
No worries.
Okay, so let's wait whether in the chat box anyone else has any comments or questions, or you can just unmute yourself and ask directly. Okay, so seems like no questions right now, and of course this is recorded, so I will take care of uploading those this webinar in about a week online, and I will share it with you, so you can of course share it with anybody else or just rewatch this nice presentation. So thanks again everyone for participating, and special thanks to Steffen and the SCIENION group for cooperation on today's webinar, and I'm looking forward for more cooperation.
Thank you. It's been a pleasure. Bye-bye.
Thank you. Have a good day. Bye.
Thank you. Bye-bye.
Bye.
Thanks.
you. Bye.
Bye.